is Xian, and I'm a PhD student at UC Santa Cruz in the Department of Biomolecular Engineering and Bioinformatics. I'm working on a project to try to improve genomic analyses by incorporating information about genetic diversity using pangenomes. I'll define pangenomes in a minute, but first I'm going to give a brief introduction to regular genomics and why current practices in the field are insufficient. Every cell in the human body contains genomic material in the form of chromosomes. Humans have 23 different chromosomes, 2 of each, one from mom and one from dad. Each chromosome is made up of a single strand of DNA. DNA is comprised of 4 different base pairs, A, T, G, and C, that are strung along the DNA molecule. The order of these A's, T's, G's, and C's along the molecule, your DNA sequence, is what we call your genome. A genome is the complete set of molecular instructions for how to create a living thing. It contains all of the information necessary to go from a single cell and to develop into a complete, complex organism. Every human being who's ever lived has a totally unique genome. Differences between genomes, what we call genetic variants, define biological differences between individuals. In 2001, the human genome was sequenced for the first time. This means that we took an individual's genome and read out the entire sequence of A's, T's, G's, and C's along the DNA molecules. Sequencing of the human genome was a $3 billion endeavor. It took over a decade of work by scientists from around the world. The result was the Human Reference Genome, a standard copy of the genome that's used as the basis for genomic analyses. To this day, the Human Reference Genome remains a cornerstone of modern biomedical research. One of the most common uses for the Human Reference Genome is in discovering new genetic variants. To give you a concrete example of this, say I have a genetic disorder. I know that it's genetic, but I don't know the root cause of it. In order to discover this, I would take my genome, sequence it, and then compare that sequence to the reference genome. I would then go through the differences between my genome and the reference, what we call my genetic variants, and try to find which ones might be biologically relevant. 99.9% of the time, this analysis works perfectly well because any 2 human genomes are on average about 99.9% identical to each other. However, in the remaining 0.1% of the time, where my genome differs significantly from the reference genome, this analysis tends to be a lot more difficult and a lot less accurate. This is a subtle but pervasive problem in genomics known as reference bias, where new samples are biased to appear to be more similar to the reference than they actually are. This means that genomic analysis tends to be less accurate for people who are less similar to the reference genome. This brings me to pangenomics. The root cause of reference bias is that the human reference genome is essentially just one copy of the genome. It represents just one person's genome with no information about genomic variants. In contrast, a pangenome represents a collection of genomes from a population and incorporates information about the genomic variants between them. My lab is part of the Human Pangenome Reference Consortium, which aims to establish a new standard pangenome reference that will be a richer and more complete representation of the human genome. I've been working on developing tools to perform genomic analyses using these pangenomes. These analyses will be less biased and more accurate than current practices. By establishing a standard pangenome reference, we can make the field of genomics more representative of the human population and more useful for all people.